This giant machine is the electron transport chain, or the ETC. This is the machine that's going to harvest all of the energy from all those nice little energy carriers that we've been building up through all these steps, and it's going to build it into a giant pool of energy that can be used to make ATP. Now, before we get into the steps of how that works, let's talk about where we are. We have been in the matrix of the mitochondria, so in this fluid kind of in the center. But the ETC is actually built onto the inner membrane of the mitochondria. Remember that the mitochondrion has two membranes. So if that's the inner membrane, this might be the outer membrane. The ETC is here, and there will be multiple electron transport chains built into this inner membrane. The electron transport chain consists of a series of proteins built into the inner membrane of the mitochondrion. So if we're looking at this diagram, here's a protein, here's a protein, here's a protein, and so on. And these proteins all work together to make this machine work. Many of the proteins in this chain are known as cytochrome proteins, and all living things have to have these electron transport chains, these cytochromes, to survive because they all have to make ATP to survive. As with any machine, this machine has to be energized or turned on before it can do its job. So let's talk about how the machine is energized. Now remember that our energy carriers, NADH and FADH2, were reduced when they gained a couple of electrons. So they took energized electrons out of other molecules as those molecules were being broken down. That's how they became energized. These little energy carriers are now going to drop off those energized electrons into some of the proteins of the chain. So NADH is going to drop off two of its little electrons into this chain. FADH2 is going to drop off its little electrons into this protein. When they do that, they become oxidized again. So they become emptied of that energy and they have to go back and be re-energized again, start the cycle again. It's the electrons that actually give this machine energy. What will happen is that the electrons will be dumped into the proteins, and then they will travel from one protein in the chain to the next. They actually get handed off. So this first protein will hand it to the second one. The second one will hand its energized electrons to the third one. This one hands it to the fourth one, to the fifth one, and so on. So it's kind of like a game of hot potato, if you remember that from when you were a kid, where you're passing an object quickly from one thing to the next, keeping that energized reaction going. As the electrons travel through this chain, they leave a little bit of energy behind. So the electrons at the beginning of the chain have more energy than the sad little electrons at the end of the chain. As electrons in the electron transport chain near the end of the chain, somebody has to be there to catch them. I mean, you can't just have electrons flying off into the inside of the mitochondrion. Electrons are extremely reactive, so they can cause a lot of problems if they're by themselves and unattended. So that's what this big reaction is about. You have to have a molecule present that's willing and able to catch the electrons as they come out of the chain. In fact, these electron catchers actually pull the electrons out of the chain, and it keeps the electrons flowing through. Let's talk about some different electron catchers. Different forms of glucose metabolism use different end chain catchers at the end of their electron transport chains. Aerobic respiration, which is what we've been talking about, uses oxygen gas, O2. This is really the reason that we breathe oxygen gas. Without oxygen gas, the electron transport chain shuts down and we die. Now the oxygen gas is actually going to break down through this reaction. It's going to interact with those electrons coming off of the chain. It's also going to interact with some hydrogen ions, and it's going to form H2O, water, as a byproduct. Anaerobic respiration is the metabolic pathway that some prokaryotic cells use to break down glucose and other sugars. The key word here is anaerobic. Using this pathway allows these cells to grow in oxygen-free environments. Now, why is it anaerobic? Well, that means that it can't use O2, oxygen gas, as the catcher at the end of its electron transport chain. Different forms of these pathways use different molecules. Instead of O2, they might use CO2. Some of them use SO4, methane, and other types of organic molecules. So now that we've energized this machine, we need to discuss what it actually does. What work does it do? Well, this machine is actually a giant hydrogen pump. It's a proton pump. Remember that hydrogen ions are also known as protons. So what this thing does is as the energized electrons flow through it, it grabs hydrogen ions from the matrix, this is the matrix in here, and it pumps them across that inner membrane into this little space over here. Now remember that there's another membrane over here that mitochondria have an inner and an outer membrane. So this space here is the inner membrane space, IMS, the inner membrane space. The hydrogens are getting pumped from the matrix into this little space. Now as this happens over and over again, a gradient of hydrogen begins to build up. The hydrogen ions are getting really, really crowded. And we learned back in chapter 7 that when molecules are crowded like that, they're unhappy. They want to diffuse, they want to move. So by crowding up these little hydrogen ions into the intermembrane space, what this machine is doing is actually building up a big storehouse of energy that can be used to do more work. The electron transport chain has been pumping these hydrogen ions into the intermembrane space, so it's been creating this giant gradient of hydrogen ions. Now these ions are pretty unhappy. They want to move, they want to diffuse, so they can do some work. How are we going to use these guys to make ATP? Well, that's where this enzyme known as ATP synthase comes in. Here it's this pink and purple thing. ATP synthase is a big machine. It's got moving parts. We'll take a look at it more in just a moment. But essentially what it can do is it can allow hydrogen ions to flow back through it from the intermembrane space back into the matrix. And as those charged particles flow through the enzyme, that energizes it. That gets its parts moving. And what its parts will do is take ADP and a phosphate and stick them together into ATP. In other words, ATP synthase phosphorylates an ADP into ATP. This machine makes ATP, and it's powered by that hydrogen gradient. Up to this point, we've depended on substrate level phosphorylation to make a few ATPs for us along the way. In fact, up to this point, we've only made 4 ATPs. That's not very many. Hopefully you can see how this method is different. This method of phosphorylation is known as oxidative phosphorylation. It's much more complex, but it's much more efficient. I mean, look at this thing. This machine uses an electron transport chain, a hydrogen gradient, this big enzyme complex with moving parts, and it doesn't just make one ATP at a time, it makes a bunch of ATPs at a time. So it's much more complex, but it's also faster. That's oxidative phosphorylation. Here's a more detailed diagram of ATP synthase, and it is flipped upside down from the last diagram. Look at this thing. It has parts that rotate around like a carousel, it has parts that rock back and forth, and it makes ATP. Pretty complex. How does this thing work? Well, hydrogens are going to flow into this thing through a channel from the intermembrane space, and as they flow in, they're actually going to kind of hop on this little carousel. The little carousel is going to rotate around, and once they've made a rotation, they're going to flow through the rest of the channel and out of the enzyme. So what this does is it translates chemical energy into rotational or mechanical energy. That rotation on the top will make this part on the bottom, which is called a lollipop head, incidentally, rock back and forth. And these parts that are rocking back and forth are the parts that will actually take the ADP and the P and mash them together into an ATP. So as long as this thing is rotating, it's going to be cranking out ATPs. We say that ATP synthase uses a proton motive force because it is supplied with energy by hydrogen ions flowing through it. How many ATPs can it make? There are estimations, but approximately 28 to 30 ATP at a time. Pretty good. Here we have the whole electron transport chain all put together. Let's go through it one more time. First, we have the energy. Energy carriers, including NADH and FADH2, are going to drop off their electrons into this chain. The electrons will flow through the chain from one protein to the next until they pass out of the other side. They leave behind their energy, which is what energizes those proteins and allows them to do their work. At the end of the chain, gases such as oxygen gas are going to grab those electrons, interact with some hydrogen, and form water as a byproduct. Now, what work did the chain do? Remember, it's a hydrogen pump. So it's going to grab hydrogen ions floating around in the matrix and pump them into the inner membrane space, forming a hydrogen gradient. Finally, the hydrogen gradient is going to be used to power ATP synthase. As these hydrogens flow back from the inner membrane space into the matrix through the enzyme, it's going to energize it, and it allows it to change ADP into ATP, which is the product we were looking for. I'd like you to complete this your turn before you come to class. In this your turn, you're going to examine this diagram of the electron transport chain, and you're going to describe what you see happening at each step. See if you can figure out what this diagram means. It's very similar to the other diagrams we've looked at. In part 1, I'm looking at this reaction right here. I want you to tell me what you see happening there. Part 2, it's a little hard to tell. I'm looking for this thing happening here. These little circles that are passing through these big purple blobs. What's going on there? In 3, I'm looking for this reaction here. In 4, I'm actually looking at the red and yellow arrows going up. I want to know what's going on there. In 5, I'm looking at this big red arrow coming down. I'd like to know what's happening there. And in 6, I'd like to know what's happening with this reaction at the end. See what you can figure out. How many ATP molecules are made through this long metabolic process? Well, most estimates say that from the energy in one glucose molecule, we can make between 36 and 38 ATPs. However, that's if conditions are ideal. If everything runs perfectly, we might get 36 to 38 ATPs. However, there are some factors that can decrease the amount of ATP that's made. Let's talk about those. First of all, remember that the exchange rate between our energy carrier molecules and ATPs are not whole numbers. There's enough energy in an NADH to make about 2.5 ATPs. And remember, there's no such thing as a half ATP. Same thing with FADH2. There's enough energy there to make 1.5 ATPs. No such thing as a half ATP. So it's an estimated value. Second, when we bring the pyruvate into the mitochondrion from glycolysis, it has to be actively transported through 2 membranes to get into the matrix. Active transport requires ATP. So in order to even get the reactants to the reaction, we have to burn a little ATP, and that takes a little bit out of our profit margin. Finally, the proton motive force is not 100%. What does that mean? Well, that means that the electron transport chain is actually leaky. Sometimes hydrogens sneak through that membrane back into the matrix without going through ATP synthase. And because they don't go through the enzyme, they don't energize it. If they don't energize it, it's not going to make quite as much ATP. So there are some costs associated with these steps. This slide illustrates the theoretical yield of ATP that we can get at each step in the process. Each NADH has enough energy to make 2.5 ATPs, and each FADH2 has enough to make 1.5 ATPs. Also, in steps such as glycolysis, we have some ATP that's made, also in the citric acid cycle. So when you add all of that together, you end up with a total theoretical yield of 32 ATPs from this process. All of these different estimates differ from one another. How much ATP do we make from the energy in one glucose? Well, the truth of the matter is that we make about 30. 30 ATPs are made from the energy in one glucose. That's pretty good. So I told you before to think of the glucose as a $100 bill and ATPs as $1 bills. However, keep in mind that that's just an analogy, that the exchange rate between a $100 bill and a $1 bill is not going to be the same as in these molecules. One glucose is equivalent to about 30 ATPs, and that's pretty good. The energy transferred from a glucose molecule into those 30-ish ATP molecules actually represents about 34% of the total energy that was present in that glucose molecule. So about a third of the energy was transferred and captured. The rest of that energy is going to be lost as heat, but even that energy isn't useless. That's the heat that helps keep our bodies warm. That's the heat produced by digestion. Now, 34% may not seem like a lot, but let's compare that to a combustion engine. The average car engine uses about 25% of the energy present in the gasoline that's burned in the engine. So we're at least a little more efficient energetically than the average car. That's pretty good. This metabolic pathway is primarily used to break down carbohydrates such as glucose for energy. However, carbohydrates aren't the only game in town. Other types of molecules, including proteins and fats, can also be broken down for energy. When proteins are broken down for energy, they have to first be broken down into amino acids, and then those amino acids can enter at different parts of the cycle depending on what kind of amino acids have been made. They can enter at the point of glycolysis, or they can enter the citric acid cycle and be broken down. Fat molecules contain a lot more potential energy than the average carbohydrate molecule. So fats can also be broken down for energy. However, fat breakdown is a little bit more complex than carbohydrate breakdown, which is why your body will break down carbs before it accesses your fat and burns fat on your body. Fats can be broken down into glycerol and fatty acids, and those molecules can enter this metabolic pathway at different points and be broken down further for energy. Whooey, you guys, you did it! You finished big old chapter 9. You should now feel confident enough in your knowledge of ATP that you can finally go out and get that ATP tattoo that you've been wanting. We'll see you in class.